Thank you for joining us for the second annual Global Summit, Global Kidney Innovations, Expanding Patient Choices and Outcomes, a collaborative partnership event presented by the American Association of Kidney Patients and the George Washington University School of Medicine and Health Sciences. My name is Brian Hess, and I am a college graduate with an expertise in biology and medical sciences, as well as a published author. I have managed kidney disease for 32 years as both a kid and an adult kidney patient, having endured by both transplants and dialysis, currently of which I am on home hemodialysis. I am honored to serve AAKP as a member of AAKP's National Board of Directors. Ever since I first became involved with AAKP, I have never felt more enabled to make a difference for others like me than I do with AAKP. Today, I am pleased to introduce Dr. William Fissell, Medical Director of the Kidney Project, who will present on Emerging Innovation in Kidney Disease with Global Implications, Bioartificial Kidney.
Hi, I'm William Fissell. I'm Associate Professor of Medicine at Vanderbilt University Medical Center and Medical Director of the Kidney Project. I'd like to thank the American Association of Kidney Patients and George Washington University for the opportunity to update you on the Kidney Project and our progress in trying to create a universal donor kidney to overcome the scarcity problem in organ transplant. So this is our moonshot. It's our goal to create a biomedical device that a patient with renal failure can use, can have surgically implanted in the patient, so the patient can walk around, make urine like anybody else, and that anybody can have one. We want to overcome the scarcity problem, the scarcity model that infests how we treat patients with kidney disease today. Let's contrast that for a moment. Think about your cell phone. It might have been expensive. You might try to take care of it, but if you drop it and break it, you're not afraid that suddenly you won't be able to make or receive phone calls. There's no issue with scarcity of cell phones. But it's totally the issue with kidneys. If you need a kidney transplant, you're probably going to have to wait. And that kidney, when you get it, is going to be very precious to you, because if it fails, you fail. You revert back to dialysis, and your overall survival and your future and your day-to-day life are all quite different. So our goal is to eradicate the scarcity paradigm in kidney transplant by creating a device that will work, that will be operationally identical to a transplanted kidney. But instead of waiting for a donor organ to be available, the transplant surgeon just goes to the shelf, pulls one down, takes it into the operating room, and surgically implants it. We're obviously not the first people to have thought of this. You can go back even to the 1950s and the 1960s, and you can see pictures of Pim Kolff, first physician to successfully dialyze a patient with renal failure, with a portable dialysis machine in a VW microbus, taking patients on tours through the Alps. But if we're thinking about a device that's going to be so small that it could fit inside a human body, fit in a body cavity conveniently like a real kidney does, a dialyzer is awful big for that idea. Those of you who have been on dialysis, have prescribed dialysis, you know how big a dialyzer is. A couple beer cans stacked end on end. It's hard to imagine what body cavity that would fit in. It's hard to imagine how it would get there. And then, on the other hand, dialysis patients don't always see this aspect of dialysis, but the physicians do, and the medical directors do, and the technicians do. That is that every dialysis treatment consumes hundreds of liters of water. Every dialysis unit has a water room. And so the concept of a therapy that requires hundreds of liters of water is a fundamental barrier to something that's implantable or portable that you can walk around with. So the whole concept of dialysis might prove to be really hard to miniaturize. So instead of trying to miniaturize dialysis, we looked to what my former mentor David Humes used to refer to as Mother Nature's 60 million years of research and development. So in the very brief Bill Fissell version of renal physiology, your kidneys, each one takes maybe 600 ml a minute of blood flow and filters about 10% of that by volume, maybe a little over 1,500 liters a day of fluid volume. And that filtrate, a huge amount of fluid, more than you or I could ever drink in a day, passes over the renal tubule. Each one of the kidney's million little filtering units filters and then reabsorbs almost all of the filtered fluid. So that at the end of the renal tubule, that original 1.2 liters a minute of filtrate, 1,700 liters a day, is reduced 1,000-fold down to 1.5 liters to 2 liters that you pee every day into a toilet bowl. And so our challenge was to try to copy this rather than trying to miniaturize a therapy where you could see that there are some fundamental barriers to miniaturization. Fortunately, some aspects are easy to copy. These cells that grow in the renal tubule, they grow in you, they regenerate in you when they're injured, and they grow in a lab dish. The kidney's filters the cellular structures that are damaged in almost all kidney disease that results in kidney failure. Those cells, you can't grow them easily in a lab dish. They don't make the same intricate micrometer scale structures that are necessary to create a filter when you try to grow them. And so our challenge was to try to create a filter like the kidney's filters and try to create a tubule like the kidney's tubules. In the case of the filter, we can build that filter structure even though we don't know how to grow that filter structure. On the other side, the renal tubule, 55, 60, 70 years since the first time a human was dialyzed for renal failure, we still don't know the names of the chemicals that cause the clinical syndrome of uremia. We don't know the chemicals that make you sick when your kidneys fail. But a healthy kidney knows. A healthy kidney cell knows. So instead of holding our patients hostage to the uncertain calendar of discovery science, where experts try to enumerate molecule by molecule what is the worst contributor to uremia, we said, well, let's just take advantage of the cell that knows. The renal tubule cell knows which solutes have been naughty and which solutes have been nice. It's the only Santa Claus membrane in the world. And what's more, if you use the renal tubule cell, you get to harness cellular energy. You get to use the body's own energy to move salt and water around, to be able to pick and choose between waste products and valuable metabolic substrates. You don't need a wall outlet or an electric cord or a tether. All you have to do is eat. So that's our challenge. We're going to try to build the structures that we don't know how to grow, and we're going to try to grow the structures that we don't necessarily have the knowledge to engineer. So that outlines what the talk is going to look like. We're going to start talking about the filter. I'll tell you the progress that we've made there. And then I'm going to talk about the bioreactor, the tubule cells, the other half of the artificial kidney. Tell you about the progress we've made there. Some of you have seen this slide before. It's been a staple of my talks for 5 or 8 years now. On the left is hollow fiber dialyzer. It's one of those little thread-like white fibers in the dialyzer cartridge, cut and then viewed by electron microscopy. This is the fine details of a device that keeps 500,000 Americans alive. But you see, when you look at it up close, the pores are all different shapes and different sizes. They're irregular, they're round, they're long, they're thin. They're all different shapes. They're all different sizes. But on the right, when you look at the kidney's filters, that elongated slit-like structure that separates one of these podocyte foot processes from the next that are wrapping around and grasping the glomerular capillary and filtering salt and water through these spaces. Those filters, those slit diaphragms, they're all the same size. And they're all elongated slots. So why is that? Turns out everywhere you go in nature where there's pressure-driven filtration, there's slots. They're in the baleen plates of filter-feeding whales like this humpback whale. And engineers like them too. In fact, here's just a snapshot of a storm drain in Sydney, Australia. It turns out, and we have to thank Andrew Zidney, who is chair of chemical engineering at Penn State, for helping us with these insights, that a membrane composed of slot-shaped pores that are all the same size will outperform a membrane of round pores of all different sizes. So that gave us an aha moment. Maybe if we craft a membrane that copies the shape of the kidney's own filters, we can achieve the improvements in filter efficiency. In package size, moving down from the 2 beer cans of a hollow fiber dialyzer, moving down from the pressure requirements of an electric motor driven roller pump. Maybe we can get down to something small enough that we could implant it like a transplant kidney. And so I've shifted the kidney's filters off to the left. On the right, you can see our silicon membranes. All we did, our only contribution to the field so far, has been to change the underlying technology that we use to make filters. We've moved away from the polymer spinning process, where the pores are formed by the thermodynamic process of solvents separating like oil and water in the plant, to a process that we borrowed from the microelectronics industry, where an engineer with a CAD program draws each pore one by one and has the shape and the size that the patient needs to let the bad things out and keep the good things in. And so on the right, you see scanning electron micrographs of our very first filters that we crafted 13 or 14 years ago. And you can see they're flat sheet filters because that's how you get silicon wafers. The pore itself is that little black crack with the arrow pointing at it between 2 adjacent blocks of silicon. And in the lower right-hand corner, if you snap the membrane and you look at it in cross-section, you can see these pores running from top to bottom. They're all the same size. They're all incredibly smooth-walled. Nothing's going to get hung up there. And so we took these filters, and with an extensive amount of engineering, computational fluid dynamics, surface chemistry, engineering processes, we crafted a housing for the filters where the shape of the blood conduit inside that housing allows us to avoid anticoagulants, warfarin, heparin, none of those. We have chemistries that shield the silicon filter from the hostile environment of blood. And so these filters work. Here's a cutaway from one of our very early experiments where we put the filter membrane in a little cartridge, just like this cartridge, in an experimental animal for a month. And we just collected the ultrafiltrate from our little tiny millimeter size membrane that we started with. And there you see a pouch of yellow juice. The filters filter just under the driving pressure of the animal's cardiovascular system. And so we scaled up a little bit. Next slide. We're now able to put larger devices in experimental animals, implanted in this case in a pig. We have a couple of different designs. different sizes, but they all share the feature of a carefully crafted blood conduit that protects blood from high shear stresses, from low shear stresses, and allows us to implement these cartridges, these filters, in a small cartridge that you can implant and leave in place for weeks or months at a time. You can see on the left the cartridge implanted in this case in the animal's neck. It's small enough you can put it in a neck. On the bottom you can see the Doppler tracings of blood flow through the cartridge after a month. Blood is still flowing, you still see a pulsatile waveform. On the upper right you can see the cartridge right at the time that it's being implanted. Small, it's compact, and you can see the little stacks of silicon filters and the blood conduits entering and leaving the cartridge. So in a relatively short period of time, we've gone from the concept that I had that we could change the underlying technology we use to make membranes from polymer spinning to silicon nanotechnology. That would let us achieve huge gains in the efficiency of the membranes, and we could spend those gains by shrinking the package size of the membrane down to something small enough we could implant, and decreasing the pressure requirements of the membrane low enough that we could do away with roller pumps and electric motors and wall outlets, and just use the heart to power the filter the same way that your heart powers your kidney. We then turned to the harder problem. I told you before that renal tubule cells, these cells that are responsible for harvesting the salt and the water and the amino acids and the glucose and all the good stuff that your kidneys filter and returning it to you and not wasting it down the toilet bowl. I told you these cells grow pretty well in cell culture. You can sample them, even with a biopsy needle, and take those cells and grow them on a lab dish. But cells know where they are. On the lower left is a page from Heptonstall's Pathology of the Kidney. That's a renal tubule cell. It's a beautiful, ornate cell. It's got this lush kelp bed of microvilli on its apical surface to expand the surface area of the top of the cell several thousand-fold to increase the surface area for reabsorbing salt and water. On the bottom side, similarly, there's little invaginations into the cell membrane that position the sodium-potassium ATPase, the chemical pump, right alongside the mitochondria that generate the energy from food, generate ATP from glucose and fatty acids. It's a beautiful, organized factory for transport. On the right is one of these cells grown by me on a lab dish. And I don't know about you, but if I was going to Walmart and I had to choose a cell, I think I would probably choose the cell on the left. The cell on the right, what we're able to grow in the lab dish, doesn't have that lush kelp bed. Mitochondria are scarce. There's not a whole lot of them. The bottom of the cell, the basolateral surface, is simplified. And so we were told early on that our idea for an implantable artificial kidney would never work because everybody knows that the cell on the left turns into the cell on the right the moment you put it in a dish. And so, being the engineers that we are, we asked, how does the cell know where it is? And so we wrote down on a piece of paper the differences between me and a hunk of plastic. I think many people might be surprised that there's a distinction to be drawn between me and an inanimate hunk of plastic. But several things are different. I can go to the gym all I want, and I will never be as hard as a piece of plastic is. And so we asked whether the elasticity of the material on which you grow the cells makes a difference. In the lab, the cells just sit in their little dish. In your kidney, there's fluid flowing over them constantly, refreshing the cells with nutrients and oxygen, pushing them, pulling on them, pulsing on them. And so we asked, does fluid flow matter? And then there's 100 other questions we could ask about the food that the cells eat, the hormones that the cells feel. And so we started at the beginning, and we just started testing variables. And remarkably, we found a huge difference in the way that the cells looked if we grew them on something soft versus something stiff. On the bottom, we see where it says 40 kPa. That's pretty stiff. And you can see that the cells form this lacy, spiderwebby network. And we all grew up learning that that was good cell culture. You can see all the cell-cell junctions. You can see all the nuclei. But that's not what cells look like in me. I don't have anything flat like that inside me. But lo and behold, when you grow the cells on a substrate that's got the stretchiness of healthy human tissue instead of the stretchiness of a window pane, the cells spontaneously form little balls and cysts. And if you leave them there long enough, they form tubules. These aren't cells grown in the gel. This isn't so-called 3D culture, whatever that actually means. This is just seeding them on a surface that's a little bit springy. And so we asked ourselves, does this matter? Is this just chance? And this is just a morphologic finding. And so we asked a simple question. Do we see the transporters that we need? Do we see the protein pumps on the cell's surface that we need? And so I've shown you this slide before at other meetings. Let me take you through it. Where it says A, that's RNA, that's expression. That's whether the cell is even trying to make the protein that we need. And you see there's two little short bars on the left. One's light gray and one's dark gray. It's almost indistinguishable from zero. And that really set me back a pace because I was really hoping on the soft one, the light gray one, we'd see a ton of expression. But it's low there. You grow the cells for a typical period of time, 10 days, not very long, and you can't see any difference. But if there's one thing that I am, it's careless and lazy. And so about a month into things, my lab manager came to me and said, Bill, what do you want to do with these cells that we've been growing for a month? I said, I don't know. Take a look at them, see what they're doing. And lo and behold, you can see there's one tall gray bar. And that's the cells that were grown on a substrate that was springy like healthy kidney tissue, where we gave them time to differentiate. We gave them time to heal from the wound of being splattered down on an artificial surface. On the bottom, you'll see there's one dark band there, and that shows that it's not just the nucleic acids, it's actually the protein itself. And when you look on the right, you can see colored in red on the top that there's a lot more of the red colored protein, the transporter that moves salt from the filtrate back into the blood on the soft substrates on the top than there is on the hard substrates on the bottom. But this wasn't good enough. This is still just pretty pictures. This isn't doing anything. And so we decided to measure transport. Now, here we hit a hiccup that slowed us down for quite a while. I just kind of breezed past the idea of soft and stiff gels. The catch is that these gels aren't very water permeable. And so you can't measure water transport through these cells, through these gels. But if you grow the cells on a traditional permeable support called a transwell, that's stiff. That's stiff, almost like hard plastic. We've lost our effect. So what we did was we said, well, look, there's a lot of smart people in the world, and some of them have already looked at the effect of substrate stiffness on cell morphology in other areas, in cancer, in wound healing. And they've traced out the signaling pathways involved in this response. And so we took a signal from them. We said, well, wait a minute. What did they find out? And they found out that a particular hormone called TGF-beta that you may know about, a cytokine called TGF-beta, really seemed to be the chemical signal that mediated this stiffness response. And so in the dark gray, we gave the cells a drug that blocks that hormone. And you can see in the left-hand cluster of graphs, when you go from pale gray to dark gray, the transport jumps up. So we were able to copy the effect of soft and stiff with a drug. And that liberates us from these cumbersome gels that aren't water permeable, where we can't measure transport. We're also interested, I bet a lot of you in the audience have heard of a drug called metformin that we use to treat diabetes. It fundamentally alters the energetics of the cell, the way the cell handles its energy economy. And we just took a long shot and asked ourselves, well, gosh, I wonder if that drug's going to help this out, because these cells use a lot of energy to move salt and water. That's why they're the cells that are hurt in acute kidney injury. They're the cells that starve first if you cut off blood flow to a kidney. And so I wonder if maybe giving these cells a little bit of metformin will help them out. And you can see in the medium gray bar, compared to the light gray bar, the transport goes up. And in the black bar, if you give the cells both the drug that blocks the hormone and you give the cells the metformin, you get even more transport. And what's more, if you go all the way to the right-hand cluster of bars, the ones that are way lower, if you give the cells a drug, like it's very similar to digoxin, that blocks the energy-generating pumps at the bottom of the cell, the transport drops. And so that shows us that we didn't just make a leaky cell. We didn't just use our TGF-beta blocking drug or our metformin to poke holes in the membrane. These cells are actively pumping salt and water from top to bottom, and that is exactly what we need them to do. And what's more, we asked ourselves, are we really sure that these cells are pumping the way we think they're pumping? And so we said, well, is there a drug for that? And sure enough, we give patients diuretics to block tubule cells from reabsorbing salt and water when we want to treat renal failure or heart failure. What happens if we give these cells one of these diuretic drugs? And lo and behold, you'll notice that the bar that's labeled furosemide, Lasix, you give these cells Lasix and they stop transporting salt and water. So again, we didn't just make a leaky membrane with holes in it. We're not measuring a mistake. We actually coaxed these cells to do the right thing on the lab dish. So we've gone again from the concept that maybe we can use cultured cells to harness cellular energy to move salt and water for us, to liberate us from the wall outlet and the electric cord and the electric motor, and use the cell's electric motors powered by ATP, powered by glucose and free fatty acids to do our pumping for us. And then for free, we get 60 million years of research and development by Mother Nature to help us pick out which chemicals are waste products and which chemicals the body needs. So I hope I've persuaded you that we're well on our path to creating a universal donor kidney. Our engineering work, spending a king's ransom on computational fluid dynamics like we were designing a commercial jetliner to design a hemofiltration cartridge, means we don't need warfarin or heparin or anticoagulants. The fact that we've made progress growing these cells and getting them to become functional in the funny environment of a laboratory dish means that we can do away with carrying around 200 liters of dialysate with us or cartridges of carbon to regenerate dialysis. The nature of our membranes blocks the traffic of the cellular and chemical effectors of the innate and the acquired immune system. The cells are protected from the host and vice versa. So we don't need prednisone or tacrolimus or Cellcept or anti-rejection medicines. And since the whole device is inside the patient, the filters, the cells, everything, we don't need catheters. We don't need those breaches in the skin that are conduits for infection. The bane of every dialysis patient's existence is access infections. we'd do away with all that. So to recap, to overcome the size challenge, we needed to create a new filter. To overcome the mass balance of getting rid of enough wastes so that somebody's healthy, we needed a way to concentrate those wastes into a little yellow juice called urine using these cultured cells. We finished the proof of concept for all of the phases of the project. We've demonstrated everything. And what remains is funding. I'd like to see dialysis machines become iron lungs, a device that was mass produced and saved countless lives until a better solution came along. I'd like to see dialysis machines used rarely and seen most often in a museum of the triumphs of technology that kept people alive. But we have barriers. Let me share with you what the NIH study sections that have been reviewing our grants think. They all agree that an artificial kidney would be a game changer for end-stage kidney disease. The reviewers comment that we started this a long time ago and we've made steady, slow progress. I mean, shouldn't this be what you look for in a grant application? A team that's been working consistently for years. They're not just a flash in the pan, and they've been making steady, slow progress. Wouldn't that be what you want? But instead, they knocked our ankles out from underneath us, and they said, well, you've been making slow, steady progress for a decade. I guess you're not innovative. I guess you're not a paradigm shift. Another reviewer says, the approach is logical. Milestones are stated clearly. Well, that's good. You want a grant that's logical. You want a grant that says where you're going and how you know if you're going to get there. Our approaches are based on a solid foundation. Well, what more could you ask than that? Would you rather have a castle built on sand? We even came up with a plan B and a plan C. The reviewer says, we're going to make progress. We're going to accomplish much of what we propose, but it's not transformative. A bioengineered artificial kidney to overcome the decades-old scarcity problem in organ transplantation isn't transformative. I don't know how you handle this hubristic dedication to intellectualism that leaves my patients, 500,000 Americans, waiting for something better to come along. I don't know how you overcome this. We've been incredibly fortunate over the years. David Humes, my mentor at Michigan, gave me a job when nobody else would give me the time of day, because here was the second-year medical resident who was talking nonsense about making a kidney out of silicon chips. Shiva Roy and Aaron Fleischman were and are biomedical engineers that I met at Cleveland Clinic, who, before I had a penny to my name, when I was sleeping on couches, heard what I had to say and put their shoulders to the wheel and really worked to get us our first and second and third grants. Roger Marchant was a macromolecular chemist at Case Western who helped us with blood materials interactions. Andrew Zidney is chair of Chemie at Penn State University and really helped us understand what we were trying to do with our membranes. Terry Conless and McKee at Ohio State. You'll notice that we've got a Wolverine, a Nittany Lion, and a Buckeye all working together. And really helped us with the nitty-gritty of what happens to a solute that enters the pore. Seth Karp is chair of surgical sciences here at Vanderbilt, and he has been incredibly generous with his time. He could have just delegated somebody to help us, but instead he spent hours in the operating suites and planting cartridges for us and helping us understand the implications of what we were trying to do. I'd like to give a shout out to the U.S. Food and Drug Administration. It's a lovely party trick to go into a senator's office and talk to the staffer and say, I'm trying to invent a medical device. I want to talk to you about the FDA, because their faces fall and they get upset thinking that you're going to ball them out. And then you say, I want to praise the FDA and tell you what a great job the Center for Devices and Radiologic Health is doing, because they set up a program called the Innovations Pathway Program. Jeff Shuren set that up a decade ago to spur innovation in this field, and they have truly been our partner from the get-go. I can't say enough good things about them. We've been fortunate to be funded by the National Institute for Biomedical Imaging and Bioengineering. That's the EB in these grants. We were funded by part of Zerhouni's Directors Initiative that Rod Pettigrew, the chair of NIBIB, piloted, called the Quantum Projects Program. Places in medicine where the application of technology could make a quantum leap in the care that patients receive. That program was not renewed. We've been incredibly fortunate that families who have been touched by the pain of kidney disease and who are frustrated by the limited options available to them have reached into their pockets and said, we want to help you move this project forward. And so we've been funded from philanthropy as well. We've been funded from gifts that bridge us over the gaps in NIH funding as we try to get from grant to get over the goal line, to get our innovation and our ideas out of the academic laboratory, off of the lab bench and into patients. I'm going to close with a quote from Pim Kolff, the Dutch physician who was the first physician to successfully treat acute renal failure with a dialysis machine. He's a remarkable man, responsible not only for dialysis, but also for much of the design of the Jarvik 7 artificial heart. And at the time of his death in 2009, in his assisted living facility, he was working on artificial lung technologies. And he commented that it's nearly impossible to obtain funding for something simple. And I hope I've convinced you that what we're doing in the Kidney Project is actually simple. A filter and a tubule to copy the kidney. He said, therefore, over the years, my laboratory has teetered along, always on the rim of bankruptcy, always having too many ideas and too little money to carry them out. Thank you for your attention.
We appreciate Dr. Fissell for taking the time to share his insights on medical innovation on artificial kidneys and how timely research and innovation can improve patient lives and society.